Today's lecture, we are going to look at the possibility of using endogenous compounds, that is, these are the compounds that are produced inside our body, and there is another class of compounds known as peptidomimetics. We will look at the definition a little bit later, and as well as oligonucleotides as drugs. So, we have just been looking at various strategies for drug design, but we want to go back and look at some of these possibilities and examine them and critically evaluate how good these strategies are. So, just to remind you, endogenous compounds are basically the molecules which occur naturally in the body. Okay, so, like hormones or neurotransmitters, and these are examples of molecules which are produced inside the body. And of course, many of these could be extremely useful because the imbalance in the levels of these neurotransmitters or hormones sometimes leads to disease. So, the question is, why not use these in medicine? Okay. So, let us start with the neurotransmitters. So, there are a number of non-peptide neurotransmitters which are present in the body, and they are very simple molecules and they can be prepared very easily in the lab. So, the major question that one asks is, why are these not used very commonly as drugs? So, the classic example is dopamine. So, we have already looked at this in fair detail. The dopamine levels need to be elevated in certain kinds of diseases such as Parkinson's disease, and so one could think of just administering dopamine so that we can maintain the makeup for the balance. So, why is this not done? So, the problem with neurotransmitters is that they have not been designed to travel long distances within the body. They are typically used with very short range signaling, and they are therefore not very stable. Okay. So, for example, if we consume a medicine or a pill containing this neurotransmitter, it is quite likely that they will get decomposed in the stomach because of the high acidity. And now, therefore, you will probably have to inject it. Okay. So, then the injection of course is going to help us bypass the first pass effect, but the problem is that it may not survive until it reaches the target receptor. So, the major problem with using neurotransmitters or the natural neurotransmitters is that they will not be stable enough and they may not survive to reach their target receptors. And one of the reasons for this is because the body has efficient mechanisms which are used to inactivate these neurotransmitters. So, for example, if the neurotransmitter is not inactivated, then they are going to continue to pass on the signal from the nerve to the target cell, and that is not very useful. So, there are endogenous mechanisms or natural mechanisms by which these neurotransmitters are going to be metabolized. Okay. Typically, they are very short lived. So, any neurotransmitter that is injected into the blood supply is going to be inactivated by enzymes, or what happens is that they are going to be taken up by cells via transport proteins, and so both of these are going to result in a loss of the amount of neurotransmitter available to reach the target site. So, even if they were not inactivated or removed, they would really be poor drugs because they would lead to many undesirable side effects. So, for example, if you want the drug to be active in the lung, it is going to be activated in all areas including the lung. So, a shortage of a neurotransmitter may be only in a small area in the brain, but in other areas in the brain it may be quite normal, or in other areas of the body it could be quite normal. So, if we give the natural neurotransmitter, then it would be impossible for us to stop an overdose of the transmitter, and it will be able to trigger the signaling in other areas as well. Of course, this is a problem not just with neurotransmitter, but with all drugs. Okay. But it has been discovered that the receptors for a specific neurotransmitter are not identical. So, we have looked at this previously. So, therefore, there are different types and subtypes of a particular receptor, and the distribution around the body is also not uniform. So, if you use the natural neurotransmitter, there is no chance for us to distinguish among these various subtypes. However, by using a synthetic molecule such as a drug, it is possible that we may be able to distinguish these subtypes. So, one subtype of a receptor may be common in one tissue, whereas another subtype is common in another tissue. So, the medicinal chemist can now design an appropriate synthetic drug which takes advantage of these difference and which will help us target a particular receptor subtype where the natural neurotransmitter would not be able to do it. Okay. So, in this respect, a medicinal chemist actually has an opportunity to improve on nature. So, the other part of it is that we also cannot assume that the body's own neurotransmitters are perfectly safe. So, for example, if one is being constantly injected with the body's neurotransmitter, then it is possible that there is tolerance and addiction associated with these drugs. So, a classic example is heroin. Heroin tolerance and addiction are quite commonly observed. So, it is possible that one can be addicted to one's own neurotransmitter and hormone. So, a parallel example here is that some people are addicted to exercise, okay, and so and they are compelled to exercise for very long hours in each day in order to feel good. Because during the process of exercise, which I would highly recommend, that it releases hormones and neurotransmitters which can produce good feeling or what is known as a high. And so there are certain types of people who are susceptible to to this sort of feeling, and they exercise more and more to achieve this high. But as a general concept, it is a good idea to exercise regularly, and it need not be for several hours, but it can at least be for 45 minutes to 1 hour. So, these people who are addicted to this exercising actually suffer what are known as withdrawal symptoms, which we have looked at as previously, such as deep depression. And so, this is the same phenomenon that drives risk taking people, for example, mountaineers, to attempt feats such as climbing very tall mountains, which they are probably quite sure that it will lead to their death. Okay. So, the thrill seekers or a thrill of a danger produces these hormones and neurotransmitters, which can again in turn produce a high. It also explains why some individuals choose to become mercenaries and risk their lives by traveling around the globe in search of wars to fight. So, the natural neurotransmitter as a drug may not be the best concept to take forward. So, unlike neurotransmitters, hormones have the potential in drug therapy because they normally circulate around the body and behave pretty much like drugs. So, we have already seen, for example, when there is a sedative that is given or when there is an anesthetic that is given intramuscularly, adrenaline is injected along with it, so that it takes time for the sedative to release, to be distributed across the body. This is for a local anesthetic. So, most hormones are peptides and proteins, and some naturally occurring peptide and protein hormones are already used in medicine. So, for example, insulin is already been very commonly used in diabetes, calcitonin, erythropoietin and so on. These are already proteins which are used quite commonly. The other problem with using a hormone is in purifying it. So, isolating and purifying a hormone from blood samples is quite impractical because there are very really tiny quantities of the hormone present. So, what is used is a technology known as recombinant DNA technology, which we have already looked at previously. So, here the human genes for the protein are cloned and then incorporated into the DNA of fast growing organisms such as bacteria, for example, E. coli or yeast, and in some cases mammalian cells. And then these cells then overproduce this hormone, and then we isolate the sufficient quantities of the protein by running this time and again. Okay. So, however, many endogenous peptides and proteins have proved quite ineffective. So, the reason for this is that they are highly susceptible to digestive and metabolic enzymes. So, there are enzymes which are present in our, as we have already looked at, proteases for example, which can cleave these peptides and proteins. Not just that, they are also very poorly absorbed because of their highly polar nature. They are quite poorly absorbed, and because of the high polarity, they are also rapidly cleared from the body. So, together all of these concepts are important in designing an endogenous peptide or protein as a drug. So, also an important concept with proteins, for example, these are large macromolecules, is that they can induce an immune response. Okay. So, since we are using recombinant DNA technology, there may be elements of non-human character to the macromolecules which can elicit an immune response, and these can cause pretty severe side effects. So, one of the ways in which we can solve this problem is that you can incorporate a water soluble polyethylene glycol on the polymer. So, what this does is that once you have a protein, you can decorate it or covalently modify it with a PEG chain, which is known as polyethylene glycol chain. So, polyethylene glycol is nothing but you have so on, right. So, this is a peg chain. So, you can take this and incorporate it into a protein. So, what it does is that it, of course, it increases the solubility, and also because it forms a layer around the protein, it increases the stability as well. Now, because the polyethylene glycol is like a homogeneous coating around the molecule, it decreases the likelihood of an immune response. Pegylation, as it is called, also prevents the removal of small proteins from the blood supply by kidneys or the endothelial system. This is probably because of the large size of the pegylated protein. So, during the filtration process in the kidney, because these pegylated proteins are quite large, they are not filtered into the kidney nephrons and remain in the blood supply. So, together pegylation is one of the important strategies to deliver protein. The peg molecules surrounding the proteins can be viewed as a kind of a hydrophilic polymeric shield, which both protects as well as disguises the protein. So, let us say you have a protein here, and around this you have a sort of a shield of this polyethylene glycol layer. Okay. So, it also has an added advantage because polyethylene glycol polymers are extremely well tolerated by the body and they do not show any toxicity. So, the enzyme adenosine deaminase has been used in a similar manner, and protein peg conjugate has been prepared, and this conjugate has been used for the treatment of severe combined immunodeficiency known as SCID. Okay. So, this syndrome is associated with an immunological defect which is because of the lack of adenosine deaminase. So, therefore, you can deliver this enzyme adenosine deaminase as a pegylated form in order to sort of increase the levels of this enzyme in the body. These conjugates, these pegylated conjugates, have much longer plasma half lives than the purified protein alone, and they are less likely to produce an immune response. The next endogenous molecule is an antibody, and antibodies we have already looked at previously are quite commonly used as drugs now, and a lot of biotechnology companies are now producing a large number of antibodies or antibody based drugs with the aid of genetic engineering and monoclonal antibody technology. So, antibodies are used to recognize chemical signatures of particular cells or macromolecules. They are also used to carry drugs to specific targets. So, here for example, we have already looked at the antibody is conjugated to a drug, and this is used for targeting a particular site, and the drug is delivered along with it. There are a number of antibodies which are used as drugs. The next major molecule to consider are of course endogenous peptides, but we have already looked at some of the pros and cons of using these endogenous peptides and proteins. So, one can sort of work around this by using what are known as peptidomimetics. Okay. So, mimetic means to mimic or to look like. Okay. So, these peptidomimetics are molecules which are look like or feel like or behave like peptides. Okay. So, because peptides continue to be important lead compounds, and a lot of medicinal chemistry has worked around peptides, and because peptides as receptor ligands or enzyme substrates are very commonly found. Okay. So, therefore, we do not want to give up on peptides altogether. So, therefore, we would want to be able to deliver a peptide or a peptide like molecule which is known as a peptidomimetic. So, consequently, drugs which are designed from these lead compounds which are peptide like in nature have to be taken forward. So, the pharmacokinetic properties of these first generation drugs of using peptides are many times unsatisfactory. Okay. So, a number of strategies have evolved to improve the bioavailability and to achieve a higher level of or acceptable level of these molecules in the blood supply. So, therefore, disguising or reducing the peptide nature of the lead compound to generate a structure which is more easily absorbed from the GI tract and more resistant to digestive and metabolic enzymes is highly desirable, and these are molecules which are known as peptidomimetics. So, one approach that is used to increase the bioavailability is to replace a chemically or enzymatically susceptible peptide bond. Okay. So, here is a normal amide or a peptide, and so these are enzymatically susceptible, right. And in order to prevent this, one could use a range of these functional groups. The first example we will take is to look at a peptide bond which is replaced by an alkene. Okay. So, here if you replace the peptide bond with an alkene, then this alkene is a bioisosteer for the peptide linkage. The olefin mimics the double bond nature of the peptide bond. So, because we have already looked at previously that the peptide bond has significant double bond character, okay. But clearly the olefin or the alkene is not a substrate for proteases, right, or peptidases, and therefore, this is extremely stable during the during metabolism, especially to peptidases. However, the peptides bonds in many lead compounds are often involved in hydrogen bonding interaction. So, you have hydrogen bonding interaction possibly due to the carbonyl as well as the amine. And now, once you remove this amide altogether and replace it with an olefin, then you are removing the hydrogen bonding donor and accepting capability, and therefore, you may lose activity. Okay. So, this possibly results in a significant drop in the binding strength. Right. The alternative is to replace this amide with a ketone, because the ketone still has the carbonyl group on it, or you can replace it with an amine, right. So, here only one of the two interactions is being lost, and therefore, it might help compensate for the loss. However, the problem with these two approaches is that the double bond character is completely gone. So, there is a greater chain flexibility also, because the amide bond is more restricted, and therefore, you do not have the same sort of flexibility. The next option that is typically considered is a thioamide. So, here instead of oxygen you have a sulfur. Okay. So, this group retains the planar shape of the amide, and the NH moiety can still act as a hydrogen bond donor, right. However, the sulfur bond being quite large and quite less in electronegativity compared to the oxygen is a poor hydrogen bond acceptor. Okay. So, this strategy would be useful if the original carbonyl oxygen forms a hydrogen bond to the active site of peptidase enzymes. So, if a peptidase enzyme binds to the carbonyl to cleave it, now by putting in a carbon double bond sulfur, you are reducing the possibility of it getting hydrolyzed by peptidase enzymes. Another approach is to retain the amide as it is, okay, but to protect it or disguise it. So, one simple strategy that can be used is to just methylate the nitrogen of the amide group. So, this keeps pretty much the same properties, but it improves the stability of the molecule towards proteases. So, the methyl group acts as a steric shield and prevents the important hydrogen bonding interaction that is supposed to take place between the NH of the original amide and the active site of the peptidase enzyme. So, therefore, now that this steric shield is present, the rates of hydrolysis significantly go down, and so N-methylation is also one way to generate an amide like or to retain the properties of the amide as a drug. Another unrelated or a completely different strategy is to flip the enantiomer that we use. So, for example, if you replace the L enantiomer that are normally present in proteins with the D enantiomer, then what happens is that this makes the relative orientation unrecognizable to digestive or metabolic enzymes, especially if that side chain is involved in binding interactions. So, here for example, you have an L amino acid, and if you convert you have two L amino acids, but if you convert one of these to a D amino acid, then it is possible that this may not get hydrolyzed as well as the original molecule. But the problem is that by making this change, we also might make the peptide unrecognizable at the desired target. We have already looked at previously that the relative positions of these side chains may become important in binding to targets. A third strategy is to replace the natural amino acid residue with an unnatural amino acid. Okay. So, here the unnatural amino acid, since it is not present naturally, there are situations where these are going to survive much longer than the normal or the natural ones. So, this is a tactic that has worked successfully in structure based drug design. So, once we have a map of the binding interactions of the let us say the active site with the ligand. Okay, so, here are the major binding interactions. Now, this is again obtained by X-ray crystallography and molecular modeling. So, once we have these interactions set up, then we can design a new peptidomimetic compound which can have the same or a similar interactions, but do not have the natural amino acid as the component of the molecule. Once we identify these binding subsites in the targeting binding site, and then what we do is we start designing molecules which can fit these subsites much better. Okay. But the major condition here is that we do not use the natural amino acid and we use what is known as a unnatural amino acid. So, what this could lead to is actually it could lead to an improved binding affinity of the peptidomimetic, because now we are adding in more components of binding to the molecule. At the same time, it is clearly less recognizable to the digestive and metabolic enzymes. So, the lead compound for the antiviral drug saquinavir contained an L-proline residue and occupied a hydrophobic site of a viral protease enzyme. So, here is the L-proline residue and it occupied a hydrophobic site. So, during drug design or drug development, this proline residue was actually replaced by a decahydroisoquinoline ring. Okay. So, not only was this little bit more resistant to hydrolysis, but it also helped in fitting better in the binding site. So, one of the problems with peptidomimetics is that they are often quite hydrophobic in nature, because we are now going to add more carbons, for example, and this can pose a problem because they are very poorly water soluble, right. So, when these molecules are to be taken orally, they are quite poorly absorbed. So, of course, what we can do is we can improve the water solubility by increasing the polarity of residues. We have already looked at these strategies in our previous lectures. So, for example, an aromatic ring can be replaced by a pyridine ring. So, here it is important that we know that this whatever change that we are making does not involve the major binding sites. Okay. So, for example, here the binding interactions with the target and the ring should remain exposed to the surrounding water medium when the peptidomimetic is bound. Otherwise, what will have to happen is that the binding site, binding group will have to be desolvated before it goes and binds to the binding site. That is, the ligand will have to be desolvated before it goes and binds to the target, which results in an energy penalty. Okay. And this would of course lead to a decreased binding affinity and loss of activity. Another potential problem with peptide lead compounds is that they are invariably quite flexible molecules with a large number of freely rotatable bonds. So, a lot of studies have suggested that such flexibility is not very useful for making the molecule orally bioavailable. So, certain rigidification tactics, which we already looked at previously, may well be beneficial. Okay. The next major naturally occurring molecules are nucleotides. So, oligonucleotides are being studied as antisense drugs and aptamers. Okay. So, here is the normal structure of a nucleotide, and so here the phosphate, for example, can be modified to a thiophosphate or a bis thiophosphate or a methyl phosphate, for example, and this sort of prevents the or improves the stability, and also in certain cases it can alter the binding. Okay. You can also have the sugars being modified, and of course, the bases can also be modified. So, all these three possibilities are in play. Okay. And we have already discussed previously the roles of oligonucleotides as drugs, and so we will not be spending much time here.